Thank you for joining us for the Dean's distinguished lecture. I'm Stan Gerson, Dean of the School of Medicine. I think we have some folks on Zoom or live video as well. We've invited two of our alums back to celebrate their 30th and 35th reunions from the class of 87 and 92, Jay Short, whom you see on the screen, and Dr. Yuling Lau, if I made that mistake. What I'm really excited about in the conversation that we're going to have with them today is the incredible effort that they have made successfully throughout their careers in commercialization of medical discoveries and breakthroughs. And it's easy to talk about discoveries and breakthroughs until you realize just how complicated it is. And you'll see from the two of them how it's the lives that they live and not an afterthought. It's how they address everything that they do. And I'm delighted that our new president, Dr. Eric Kaler of the university, has made advancements in breakthroughs and research discoveries as a major top priority. And he's allowed us to consider how do we make those joint, both academic, strategic, and financial investments in this space. So getting used to bringing forward our new concepts, both in diagnostics, discoveries, therapeutics, and the like, will really help advance, I think, our entire field and the mission of the university and of the School of Medicine, certainly, which is to bring advances to human health here and across the globe. So we're going to learn a lot about both the careers and the way these folks think. So I'm going to tell you first about Jay Short. Jay received his BA in chemistry from Taylor University and then came to Case Western, where he did his PhD in biochemistry. He reminded me that he was quick to the gun and did his degree quickly, so quickly that he actually had to come back a year later and defend it. And so he left us in 85 and received his degree with the class in 87. He worked in biochemistry with Dr. Richard Hansen, who was chair at the time, who had assembled an incredible group of experts in biochemical pathways, protein synthesis, intermediary metabolism. And on the other hand, Jay wasn't alone. He had friends that are in the audience today. Tony Winchell-Boris, who's now our chair of genetics and genome sciences, and Maria Hatzoglu, who's a professor in biochemistry as well. And last year, Maria was honored by the 2021 Case Medal of Excellence in Health Sciences Innovation, which I was delighted to surprise her with just about a year ago. So I got to know this crowd because I was a young assistant professor. And in 1985, a paper was published in Science on genetic engineering of hematopoietic stem cells, a topic about which I knew absolutely nothing. And so I asked Richard Hansen if I could join the lab, come over from my appointment in medicine, and spend some time while I had my lab doing conventional things. Came over and learned, and I needed some friends, and my friends turned out to be Jay, Tony, and Maria. And a young postdoc named Ink-Young Lim that Maria and I advised. And Dr. Lin became the dean of Jeju University in Seoul, South Korea, and just has retired. So those overlapping lab experiences both taught me a huge amount. And I remember our lab meetings in Richard's office, with him looking down at his scribble pad with the colored pencils and putting out doodles, which must have been some form of his interpretation of biochemical pathways, while we were discussing our blots and next crosses and the like. And they, for those of you who frequent biochemistry, are now sitting on the walls of the Department of Biochemistry. What was interesting, on the other hand, even as fast as that lab went, Jay was working faster and harder in biochemistry, and immediately jumped to the West Coast into biotechnology, something which is perhaps common now, but certainly not common then. He went to work at and then became president of Stratasite and then vice president for research at Stratagene, which then transformed into Agilent. We bought his products, including a mouse that he invented called Big Blue, which allowed us to do a lot of genetic analysis of mutations. And he also hired a young staff technologist named Jane Reese, who about 5 years later arrived in Cleveland, and I hired her as a technician, and she's now the incredible lab director for our National Center for Regenerative Medicine. So as you can imagine, Jay has had a huge amount of influence on all of us here in Cleveland. So what's Jay up to now? He's the CEO and chairman and co-founder of Bioatla, a San Diego-based company, but that doesn't prevent him from spending some time in Wyoming. He's also chair and co-founder of Himalaya Therapeutics, chair and founder of Inversigen, and he's going to tell you about what these things do, but also has time to be a founder and former president of the E.O. Wilson Biodiversity Foundation and the Half-Earth Project. So he's been a good citizen of the world, as well as in the space of biotechnology. All of that activity, I told you he wasn't a slow mover, has led to over 500 issued patents, and he ranks pretty highly on the MIT patent score. So I want to move on and let Jay give us some impressions about how we got to where he is today. Jay.
Well, that was really kind, Stan. I really appreciate it. It's so wonderful to be back here and to see so many good friends and have this opportunity. I want to make sure everyone can hear me. Okay, yeah, I see some nods. As we get older, our hearing dies off, and partly from driving tractors and flying airplanes and things like that. But it's really great to be here. It's an honor to be here, given how much success many of you have had. This university is admirable around the world, and I've seen that over and over, that it's been an asset for me to have come from here, and I'm really grateful for that. So I was asked to give a little bit of, information on our story around commercialization, some of the influences, how my career evolved, and of course at the end, who are some of those individuals that were instrumental along the way. And as I thought about this, it really was impossible to deconvolute the two first points, because they really are interact together, they're entangled, there's no way to really, one leads to the next, and you'll see that, I hope. But as with any story, it's important to go back to the beginning. And so I'm a farm boy from Indiana, lived 2 miles from the Ohio line. We just bought our most recent farm a year ago in August, so we still keep it and we still grow it, and my mom's out there. And so it really allowed me to have an appreciation and understanding of commodity businesses, agriculture, entrepreneurship, and but in addition, there was a twist to it. My dad was a chemistry physics teacher. My uncle's a cardiologist. My other aunts and uncles were science teachers. So we had a real link to science all of that time. My dad used to take me to science fairs, and this is actually my son. I took to a science fair in San Diego, and he won that one. And this is my father next to him. So, and of course, I was a chemistry major before I came to Cleveland. So it's these kind of things that gave you a sense of science, and I wanted to go on that route. And so with this kind of raw material. And the other thing I'll just mention is that it's a lot of hard work to do a degree, and farming is a very good taste of what hard work is, and it helped a lot to be fearless in that front. So I'll populate this here briefly, but basically came to Case Western, was really fortunate to land in Richard Hansen's lab. They actually sent out a questionnaire as a grad student. Which lab do you want to be in? And at that time, I had made a decision that I wanted to be in Richard's lab. So you had to pick 3. And so I made his 97%, the next one 2%, and the next one 1%. So I was able to get in there. So you got to be fearless if you know what you want. I didn't think it would be appropriate to say 0%, but might have been. And so I originally was recruited in the macromolecular science department. Got to see polymers and pChem of polymers. And, wow, this DNA biopolymer is much more interesting to me than some of these other things. So I flipped over to biochemistry, and that was a very good move for me personally. And of course, some of my colleagues in those days, I mean, this is Tony in the middle, and David Luce was a postdoc in Richard's lab, and we went on some fishing trips, and you know, we had success, and I think it was David maybe, or did you catch that, Tony? You caught it. And so that saved face. We got 1 fish out of the whole thing. Needed a lot of rice to go with it, but it worked out pretty good. But you know, success comes in all shapes and sizes, and they can grow as you grow. And so I think that's kind of a start small and keep working up. But before I jump into some of that, you know, I thought back, what were those experiences from a technology side and then business side that I gained while I was here at Case? And it's really just a laundry list of things. And you'll see in some of the products and the commercialization that these really laid out for me or gave me the experience I need to jump into certain areas that ultimately led to commercial products. Whether it be sequencing, it was much harder then. There was no PCR. We should remember that. You had to make your own labeled nucleotides for any kind of sequencing back then, and many other factors. You'll see them here in transgenics and lambdaphage all played a role. In addition, I got my first patent here with Richard and putting Pepsi K into pigs with Tom Wagner at Ohio University. And then briefly, we had a little collaboration going with Ohio for nitrogen fixation. It's certainly eye-opening, you know, as a grad student, and you hadn't been in that area. It was kind of nice. And then I also decided to I love photography, and I decided to, and a lot of professors were having trouble getting what they wanted in the images. They, you know, the department down there didn't understand the science, and so I knew how to do this. I said, okay, I'll use the darkroom, and I'll make photos for their papers, and that's what I did. And I got a little windfall because all of the photo paper and films and everything were thrown out because they had all expired. But they were stored frozen. So I went out in the trash can, dug them all out, recycled them, proceeded to sell them back to the department for the next 4 years. And so it helped me supplement my income. But it was fun to do as well. And of course, as painful as these things may be, I think writing grants and so forth was a real power that Case helped out. And when I landed at Strategene later, it was key because we had a lot of SBIRs to write. And you know, the first week I was assigned to get a grant there at the company, and it turned out to be smaller than I even realized. But I had turned down this great opportunity to go to Joe Goldstein's, Michael Brown's lab as a postdoc, and Richard had worked to help me get that, and that was great. But I took the risk and jumped to biotech out in San Diego. I always wanted to go to California, and so. In the next slide, I'm going to just touch on some of the cloning systems, as well as this genetic test that Stan mentioned, the polymerase, and ultimately this antibody repertoire there in terms of some of that commercialization. And, you know, I think the challenge back when this is pre-human genome days was getting those libraries built. In fact, we had to build new cloning systems to make these things work well. And we were doing some of the first human chromosome sorted libraries that came out of DNA out of Lawrence Livermore. We've actually built the libraries that led to the muscular dystrophy gene being cloned. But it really required more efficient systems. So one of the things was to see if we could take an M13 origin, split it apart, put it inside a lambdaphage, such that the lambda vectors would propagate normally, but when you put a helper phage in, it would just automatically subclone that gene and then allow you to manipulate it much easier. And I built all this when I first got there, and it didn't work at all. It was terrible, and they just had fired the other PhD person that didn't get their project done. And I was the first PhD scientist at Strategene in those days. And but what I did is I ended up grabbing a helper phage and mixing it and said, okay, because originally we put the gene 2 protein, for those that know, inside the cell, but that became incompatible. It's too much clipping and so forth. But by bringing in the phage transiently, it worked. And so I survived the first shot, and we ultimately set up our first custom transgenic animal facility in the United States, and we offered it to people, and we built mice and rats and so forth. But it wasn't too long till it made sense to, why don't we put those two things together? Why don't we put lambda-phage with that various inserts in it into a mouse, and if we just isolate the DNA from any tissue and mix it with lambda packaging extract, you could plate that out, calculate the number of blue and white plaques, and get your tissue-specific mutation rate. In addition, you would have the sequences so you could know if there were transitions, transversions, and you would get that particular profile. And we studied everything from mutagens to aging, and it was, many surprising results. But this ended up being, and now it's an FDA qualified genotoxicity test that many companies use around the world. But the other interesting thing is we were working out of Yellowstone and with other high temperature polymerases because PCR was emerging and there was challenges in terms of fidelity. And we had a proofreading enzyme, and the challenge with that was the mutation rate of any DNA you just isolated from anywhere already had a baseline mutation rate that you couldn't measure what the fidelity was. It caused a lot of background. Well, in the tissue-specific analysis of mutations, we learned that germ cells have about 1/10 the accumulated mutations that other tissues have. So it turned out that that particular source of DNA could allow us to drop the background so we could measure the actual mutation rate. It turns out that's an assay that almost everyone's using these days for high-fidelity measurement of PFUs, or of any kind of polymerase. And then, of course, the next step was recombinant antibodies. And this led us to create a subsidiary called Stratocyte. And we had done a collaboration. My lab did a collaboration with Richard Lerner's group out of Scripps. And we built the first combinatorial antibody library, so allow you to PCR the whole antibody repertoire, put those in vectors, and then screen directly. Ultimately, this technology was transferred to Cambridge Antibody Technology, but it allowed us to really to move forward in terms of an area of expertise that allowed us really kind of an intro to medicine in a way, because we're doing a lot of collaborations in this. Through this period, go from a staff scientist to head of research and head of operations, but then really importantly, become president of this subsidiary. So this shifted my career and allowed me to move on to other things. And the next company was Diversa. And in this one, working in Yellowstone, and some of you may know I live in Jackson Hole most of the year now, but some of those early relationships came from Strategene and Diversa. And what we intended to do there was to say, can we harvest DNA from the environment directly and be able to express, whether it's archaea or bacteria, high temperature, low temperature, halophiles, psychrophiles, doesn't matter what it is, can you express those heterologously and high throughput with assays? And we were able to accomplish that. And one of the key reasons why is because we used lambda-phage. The lambda-phage would blow up the cell. If you tried to maintain some of these genes and express them stably without those kinds of approaches, you end up losing a lot of that DNA. It just the cell can't handle it. But by having it grow 20 minutes in lice, that was fine. And that opened up that whole field. And I'm going to give you a couple of product examples that came out. There were many products that were commercialized. And protein evolution was the other piece that we added on to that. And I won't get into that too much other than let's look at one of these products here. This was a deep sea ocean vent material, and we had isolated a thermostable enzyme with low pH. And the concept was, it didn't matter, but a lot of people were making ethanol, but there's other applications. But the cost of the enzymes were largely prohibitive for a lot of things. And so the idea was, could we get a high temperature enzyme and have it operate under low pH, be able to put it into grain, like corn, and allow that enzyme to be expressed there so it could process the starch. And actually it worked amazingly well. We did a collaboration with Syngenta, who actually did the transgenic crop area. And you just simply heat the kernels, stain for starch, and you can see this, the transgenic grain on the right-hand side, processed all. In fact, it was so efficient, you could take one kernel of that hydrothermal enzyme grain, put it in a bushel of regular corn, and will process the whole bushel. And so all of a sudden, your enzyme costs are zero because you're still buying the corn. There's no manufacturing cost. It's there, other than what you would pay for corn. And it ended up being a very large product with really, I think, approaching 1,000. I don't know. There's a huge product. I don't remember the amount now. And now, more recently, it was shown to increase efficiency of animal feed. So you could use an animal feed and also get all of these things are FDA approved at this point. Another example was this Phytase, where we evolved adjusted the pH, and we evolved the thermal stability of a Phytase. And Phytase is important because animals need phosphorus growth, especially monogastrics like chicken and pigs and humans. And the point is that if you take plant matter that has plenty of phytic acid, plenty of phosphorus, but the phytic acid molecule that is structured in prevents that phosphorus from being available for bone growth. So what do farmers do? They buy the grain, they feed it, and then they pour in phosphorus on top. So you get not only the phosphorus from the plant material, but you got it from the additional material. The problem is you go downstream and it goes into the rivers, and you get cyanobacterial blooms, you get red tides. And so this started to become regulated in Europe early on at this time. And so this just shows you by simply taking our Phytase, adding it into the feed. You have to pelletize it, so it has to be really stable and has to have the right gut pH stability and so forth. But you can see the growth difference of simply adding the enzyme versus the best at the time of a competitor enzyme, which was Novo Nordisk at the time. And Danisco had this, now DuPont owns it today, and it's a multi-$100 million product, and but while also in cleaning the environment. So this kind of between the experience at Yellowstone and around the world, we had probably 2 to 3 dozen relationships around the world, between Kenya to Yellowstone to Costa Rica to Russia, for accessing biodiversity. Each of those looking for unique environments. But as I left Aversa, I decided that the link to biodiversity was very interesting to me, and I wanted to maintain it in some fashion. So I decided to start a foundation. Ed Wilson was an advisor at Diversa. We admired a lot of his work, and so I asked him, I said, hey, look, I'm going to start a foundation. I wondered if you might want to be an advisor for it. Long story short, we ended up naming it after him, which was, I think, a great idea. And he's now they've done very well with that. I stepped out after a few years. I realized I belong in companies more than I do in these foundations, because one asks for money and one builds money, and they're two different things. But we had wonderful experiences there. We had Harrison Ford on the board, and Paul Simon just donated his concerts to proceeds for his concerts for this foundation. It's done very well, and obviously doing much better that I'm not leading it, and I'm grateful that it's just continuing on. But I want to now move on to Bioatla, and it's a cancer therapeutics company, and Diversa had decided to go on to ethanol. We had done antibody work here, and obviously an extension of what we had done in strategy. And when they decided they were going to get out of that and just do ethanol, they were laying off the people. And I said, so I'm talking to my wife, and I go, well, maybe we should start an antibody company with our old people. And she goes, that's a great idea. So we did that on a kitchen table. So we decided, let's fund it ourselves. Let's see what we can do. And we can maybe do services, build antibodies for other people until we figure out a technology that we think is differentiated enough in order for us to go into integrated drug development. And so it was on the hope that we would get that. We ended up making 120 antibody therapeutic programs for other companies before we got finished figuring out what we were going to do. And they were more like traditional ones. And just as a footnote, Himalaya is a spin-out. It owns, which my wife is president of, which is owns a lot of our drug rights in Greater China. And let me talk so just for 3 or 4 slides about the technology, which is certainly exciting from my perspective, that I'm very active with. And I think you'll see the link over time between environmental conditions and how we got down this path. But I think, as most of you know, cancer cells are acidic. They're acidic on the outside of their cells. I think we're familiar that PET scanning is a great way to detect cancer. What are you doing? You're labeling glucose that gets uptaken by those cells. And you can ask the question, whereas in contrast, blood is always pH 7.4. I mean, even acidic acidosis, physicians intervene around 7.2, 7.1 because you're dead at 6.9 if your blood goes down that low. Most normal tissues are above pH 7.4. So the question was, could we build antibodies that would only work in acidic environments but keep them off in alkaline environments? And this is a very tiny window of pH shift. And you could ask the question, why are they acidic? Well, in order for a cancer cell that's constantly replicating, it's duplicating the lipids, it's duplicating the nucleic acids, many of the proteins, amino acids. So it's a lot of those precursors require glycolysis to drive. And the last step is lactic acids, which gets secreted. And so, and this is a cellular phenomenon, so more than a tumor phenomenon. So what was interesting is that we originally went into this thinking if we could get 2 to 3-fold improvement in therapeutic index, what an amazing advantage that would be, because the big problem is it's hard to find targets that are only on tumor cells but not on your normal cells. And I think we all know when you're treated with cancer, it's a race between killing the cancer and killing yourself. There's too many side effects in a lot of these drugs, and you've got to have potency. So, but we made a surprising discovery that there was a whole class of small molecules naturally occurring within the body that in normal cells bound to they're negatively charged, and they bind to positive charged amino acids on all of your proteins. And generally we think about this of water molecules sitting on these kinds of charges, and it's hard to push them away, too many kilocalories. But these molecules were able to push them out of the way. So the kind of way I imagine is like you have a snowball and you put pepper all over it. These things are everywhere. I mean, it's really covered. And so what kind of molecules are there? Bicarbonate, the hydrogen sulfide, there's others. There's a class of them. But what happens when you go into a disease environment like cancer, shift to acidic pH, hydrogen ions displace these molecules. And so all of a sudden now you have all of these small molecules flying off of these positive charged sites, and you create these novel epitopes, and you're probably creating a lot of interactions that are not all good between those proteins and their ions than what else is out there. So the thinking was, could we make antibodies directed against these different sites? And the reality is it went the other way around. We actually built the antibody that had selection and then worked backwards to understand what happened and why it was so good. But it took us a while to figure out. Ultimately, we found that the vast majority of these small molecules, we call them protein-associated chemical switches, or PACs, were on the antigen side, and that their antibodies were going in against those sites. Now, the reality is they're all proteins, so they can be on both sides. And so you could make them so they're both sides. You can get the product of the two working together. So now on the next slide, I'd like to show you what this can do in terms of giving you antibody a therapeutic selection. The top left-hand figure shows 2 mice in each panel. The left one contains a human tumor. It's immune-compromised animals. The right one shows you normal tissue, human skin. And you can see that the approved drug, cetuximab, can't determine the difference between which one is a cancer cell and which one is a normal cell, even though they all have this EGFR. However, if we build what we called CAB, or conditionally active biologic antibodies directed against these sites, it leaves the normal skin alone. And so this one was our first one, had a 12.3-fold improvement in therapeutic index. We subsequently now have built a bispecific molecule which enters the clinic later this year, and we have a 160-fold therapeutic index improvement. And how do we do that? Because we have a EpCAM on one arm, we have a CD3 on the other arm. We'll get the selectivity of both arms. You need both arms to be effective. And so now we have 4 therapeutics, 2 in phase 2 that are in the clinic for many different indications. And this is just one quick snapshot on some human data that came out. In phase 1, we did an all-comers trial for this drug, ROR2. It's an ADC, antibody-drug conjugate, CAB antibody. And the first patient had failed both PD-1 and CTLA-4 independent and combination checkpoint inhibitor therapy. Really only had a few months to live, and this was in a lymph node. That's why it doesn't go down to the baseline, but it's a complete response on that patient. And we just got our first one in phase 2 in earlier this year. We got our second patient in, and also another complete response. And we have our third patient in. We'll see what it reads out here in the next few months. But it's kind of intriguing and very encouraging data across lung cancer, sarcomas, and ovarian, and many other areas that we're going. So it's a very exciting for us technology, but this next program, which kind of stems from this, is probably the last program I'm going to do if I fail. And of course, if you look historically, this is a picture of me at Case down here in the hood after Tony showed me how to do tissue culture, and then getting my diploma. And this is on the car ride out, from Indiana. I just left Case and heading out California. Coming over here, we're diving. There's these underwater spires in Yellowstone at 20 to 30 feet under the water. We were scuba diving to pick up pieces of this silica because of the microbial consortium that was there. And this is up in Mount Kenya. And from there was the O. Wilson Foundation, and then Bioatla starting. And then I was doing biomass conversion to protein and also coal conversion into methane, which I'm not going to talk about. And then leads us to the last one. So, I'm aging through all of this, and it seems to be accelerating. And so, and this is me as a freshman in college, and so I'd like to see if we can get back to some of this. And if not, well, then that'll be it. But I want to say, what's the basis for this company that we started? We spun off of Bioatla. And it really comes out of a lot of the work at Bucks Institute, Mayo Clinic, exciting work on senescence. And this shows you the 2 similarly aged mice here at the bottom left, but the one at the top has had their senescent cells reduced with senolytic therapies. And as many of you are aware, senescent cells don't divide. They're kind of different from your normal cells. They're different from your cancer cells, which always divide. But when this paper came out, I go, wow, that's really intriguing. I wonder if these things are glycolytic. I wonder if they might, even though they're not dividing, so I was a little doubtful, maybe they're not, but I started looking for that and realized that, wow, there was hardly any literature at that point, but they are glycolytic. And I go, okay, so they're going to be acidic. They're not going to be as acidic, perhaps, as a cancer cell, but they're going to be acidic. And that means I could probably take that selectivity and potentially push that into not just taking out every cell, because you don't want to take out every senescent cell. You have heart muscle, and those senescent cells are doing work. What you want to take out are the most hyperinflamed ones. But pH is a nice gradient for that. The most hyperinflamed are the most acidic because they're trying to generate all these cytokines and proteins to get expression. So only part of the story here, but we went through and we looked through all our cancer targets, and we happened to find 3 that were overexpressed in senescent cells. And one example is the far left. So we've now built CAB antibodies to this. You can see the pH profile, blood pH going back up. And cancer can go from about 6.8 down to 5.8. I think in the inflammation area, you're really talking more 6.5, 6.8, in that world. Anything below 7, and you can see this has been designed exactly to start activity below 7. But as you move on down. And so we have our first data coming in before the end of the year on this, so we'll see what happens. And then the other little last kind of data slide I'm going to show shows you that in my family, my grandfather and my father had Alzheimer's, so it matters a lot. So I was looking through a lot of these interactions and I said, wow, they look a lot like these PAX molecules would fit in. So we went in and took a look at this in terms of ApoE3, ApoE4, which is ApoE4 has got a higher AD risk. And when you add the PAX in, you see about a 4 to 5-fold impact. What do PAX do? They reduce the binding. They're shielding interaction. And so and there's no studies out there without the packs. We put it in an abstract in July. Got 0 calls on it because I don't think people are thinking about it maybe necessarily. But the other interesting thing, we do see a correlation that, with ApoE4, it's just circumstantial at this point, but quite interesting. So because of this, what we've done, and this is one thing you can do when you're running your own company and you spun out your own company, you can make decisions quickly and they're at risk. But we've now built both A-beta, and it was encouraging to see some recent encouraging data in that world. But we also, I think a primary target is a CAB anti-ApoE antibody, because you really don't want to shut off ApoE in your bloodstream. You need it to carry lipids. But where you might want to shut it off is where it's hyperinflamed. You cross into the brain, say, that's where I don't want you working. That's where I want you to shut off. I don't want you binding things. And the idea is, can we make a very safe version of this drug and be able to keep it off in these other areas? So, for the future, so when I look back on all this, is there any common theme? It's hard to find, but at least they do involve proteins, some aspect of it. And I would say more recently, metabolism becoming increasingly important. Do we understand it? I think you can go into T-cell metabolism, you can go into many other things. I think metabolism is a huge factor, and I think this glycolysis is a fundamental driver across so many diseases. And so I have great hopes for the future of biochemistry here. And then finally, I'll just end with a few slides. I think cannot say enough about my mentor, Richard. He was incredibly enthusiastic. Everything was possible with this guy, which was perfect for me. And he was also a bit hands-off, which maybe not good for everybody, but was excellent for me. And I really think fondly of those days. And it was great to come back here to see him, and we really miss him. I think also, I remember the great times in the lab and the celebrations. I think that might be Tony, and I don't know if that's Maria there, but there's other people you will recognize, John Monahan and Bill Merrick there. But it was a great place to be. But also, I think if you look at the people you're able to tap into, Harlan Wood and Paul Burke came back to visit, and Murda Utter, who told me I didn't know much about biochemistry when he was right. Many important feedback, things that drove you to do better. See if you could drive things, and many of these people are recognized. So I'm very grateful that the department has collaborative nature. And then finally, just one slide on my father. He also built DNA products, and he built the DNA dipstick that many of you may have done before it was automated, and then anti-arc electroporation. So anyway, look, that's what I got to say. And there's so many people who acknowledge it's not possible, so I'll just give you the institutions. So thank you.
Here we go. So we have a bowl. I'm going to leave it up here just so you don't drop it on the way
Yeah,
down.
thank you.
So it says Dean's Distinguished Lecture, J.M. Short, PhD, October 7th. So great. We're going to leave it up here. Thank you. We're going to have discussion after. So I know we'll put you back up. Sit in the chair. Thank you, Jay. So that was pretty remarkable. Talk about a hard act to follow, but I have one person in the room who can follow that act, and that's Dr. Yuling Liu. Dr. Yuling likewise has been a serial inventor and entrepreneur and knows how to take concept to incredible deliveries. So I'll tell you very briefly about him. He was originally trained for his bachelor degree in chemistry in China at the University of Science and Technology, and then came here for his PhD. His thesis was on recombinant nerve growth factor, and his thesis advisor was Professor Ken Neat in the Department of Biochemistry. Dr. Liu also quickly left academics into the world of life science startups. First, and perhaps even second, as a scientist. One of his companies that he worked at was Exelixis, now publicly traded, and it just so happens that one of our board members of the university, Jeff Dewick, was the CEO of that company and chief scientific officer. And since Jeff is now on the board and chair of our finance committee. Yuling, your future's pretty bright here, so we're expecting you to come back in that setting. You will see from Dr. Liu that he's really been inventive in asking difficult questions and finding simple answers. And although he only has 30 patents, his citations from his publications have been cited over 1,500 times with an H factor over 50, which puts him well above most of us in academics in general, and certainly our faculty here. So he's found time to make an academic impact as well as being very active in the biotechnology space. He was co-founder of Panomics, which had previously been named Genospectra, which was acquired by Affymetrix and helped develop the array technology that many of us use in the laboratory. He was then vice president at a company called Functional Genomics, and then a chief scientific officer. He became the founder of his own company, the first one being Advanced Cell Diagnostics, and helped to launch and commercialize that company, which was then acquired by Biotechnique in 2016. So currently, he's on a new initiative called Alamar Biosciences, a startup company looking at immunoassays and antibody technologies designed to improve our ability to use antibodies in diagnostics as well as in therapeutics. When I visited him at his new expanded company offices, he commented to me that the problem with peptide studies and protein recognition was pretty simple. It was signal and noise, and most of us studied the signal, and he figured, well, I'll study the noise, because if you can figure out how to reduce the noise, the signal becomes easy to look at. Again, a simple solution if you can only figure it out. So without further ado, Yuling, come and tell us about yourself.
Thank you, Nick Gerson, for the invitation. It's such a great honor for me to be here. And it's wonderful to see my childhood friend, Lin Mei, who is a chair of neuroscience department, and running into professors during my time when I was doing my PhD, Professor Nielsen and Professor Samuel. And you know, if you look at my resume, basically as Dean Gerson introduced, my entire career is in the startup companies. And so it's really a pleasure for me to have a chance to share some of my experience and learnings in the startup. And I decided to maybe just use my last company as an example to highlight maybe a few points about how to make a company successful. And so my last company, Advanced Cell Diagnostics, was founded in 2006 and was acquired by Biotechnique in 2016. The company is considered successful for a number of angles. The first is that we have pioneered spatial biology and developed a groundbreaking technology called RNAscope, which enables a single molecule RNA detection in single cell, now become the gold standard technologies. It has so far generated over 6,000 peer-reviewed publications. Last year, it was 1,700 and continue to grow. It's starting to make an impact in both research as well as in diagnostics. The company was also successful financially. We spent about $25 million into this company from venture funding. And we sold the company for $325, generating $300 million shareholder values. So as some of you may know that if you get 10x return on investment, it's considered a home run. So we did hit it out of the park. And the other thing is that the company was acquired by Biotech in 2016, and the company continued to do very well. And they are also very happy about it. But you probably don't know that when we started this company, we're not exactly doing what we're doing now with the company. And so at the time, it was 2005, I become interested in the concept of circulating tumor cells. And I thought that, at that time it was very early. Not very few people appreciate the concept of circulating CTCs. And I thought, oh, this could be really interesting because, you do need to have live CTCs in the blood in order for it to invade into the tissue and cause metastasis. And it's the metastasis that's caused the eventual death. So it could be useful for therapy guidance, useful for recurrence monitoring, and potentially even useful for early detection of cancer. But CTC detection is very hard because you only have like one CTC in over 1 million blood cells. So it's kind of like searching for a needle in the haystack. And at the time, there were only two technologies. One is immunofluorescence staining based on antibody-recognized cell surface tumor antigens. And the other technology is called the real-time PCR, trying to measure tumor RNA in the cells. But both suffer significant false positive problem. As a result of that, they need to enrich the CTC by over 1 million-fold. So, and the other thing is that you cannot distinguish the live versus dead CTCs. Presumably, only the live CTC that would cause metastasis. So that's when I thought about, well, why don't we look at the RNA in situ at the single cell level? Because if that's the case, then you should be able to tell the tumor cell easily from blood cell. Presumably, you will have hundreds of copies of tumor-specific RNA in the tumor cell, but maybe at the best, 1 or 2 copies of those RNA in the blood cell. So you have substantial improvement in signal-noise ratio. So then the question becomes, how to turn a biological problem into a technical problem, which is detecting the RNA molecule in situ with high sensitivity and specificity. And that's where my light bulb moment coming up, where I say, well, why over the last several decades, people have not been able to detect RNA with high sensitivity and high specificity, and the workflow is so complicated. And there are a variety of ways to try to boost up sensitivity by increasing signals. And I thought maybe signal is not the problem. Maybe it's the background. And that's where I got excited. Well, if it's the background, then maybe there are ways to solve it. And then I came up with these double Z ideas, where it requires 2 independent probes that have to bind to a target gene of interest next to each other at the same time in order for signal to be generated. And the chance of a 2 independent probe binds to a non-specific target, which is millions of copies in the cell, are close to 0. As a result, we can achieve selective amplification of signal in the target of RNA, but not in the non-specific target. And with that simple idea, I got really excited, and I believe in it. I decided to start up companies. And I wrote 2 grants, and one was SBR grant, phase 1, and then the other one is DoD concept grant. And both of them, I have no data, just the simple ideas. And fortunately, the grant reviewer both liked the ideas and gave me $250,000. And with that $250,000, I was able to recruit 2 bright scientists from academics. I was telling them, I got 1 year of money, and here's the idea. If it works, I think we can go somewhere. If it doesn't work, we'll go home. And sure enough, it worked. It worked beautifully. And we're able to see those single punctuated dots, which is likely to be single RNA molecules. So when I first saw those images, I was, that was an incredible moment of excitement. We feel like we did it. And this is like seeing the star in the galaxy. And that's why we named the technology RNAscope. And soon after that, I was introduced to an investor who is the founder of Morningside Venture, which is one of the top tier VC in the world, in the US, and Dr. Gerald Chan. And Dr. Gerald Chan is noted for his donation of $350 million to Harvard School of Public Health, which now named after his father, T.H. Chan School of Public Health. And I remember I was meeting with him at the JP Morgan conference in a breakfast meeting. And I was explaining in a few slides about the technology, the data, and the potential of using this technology for detection and the characterization of circulating tumor cells. And about 30 minutes, and he said, I got it. I understand the technology, and I can appreciate the potential market opportunities. And I say, how much money do you need? I say, I think I need about $5 million. He said, okay, I'm ready to invest. And I have to say, okay, we're not ready. We have not built the business plan yet. So after building our business plan, and in a few months, we met again. And he and his team still like the idea and put the $5.5 million into the company. That's the start, the formal start of building a company and scaling up. At the time, he's the only investor that I met, and really lucky that at such an early stage of company development that he had a vision and took a huge amount of risk in the company. Extremely grateful for his support. And soon after that, because of this funding, we're also getting the phase 2B, the bridge grant from SBIR, $3 million. And with that money, we're able to start a clinical study, setting up a number of collaborations with UCSF, Cleveland Clinic, and University of Vermont. We're able to isolate CTC, detecting CTCs with RNAscope technology in over 150 patients. It works. It works beautifully like this one, where you can see a single circulating tumor cell in a vast number of white blood cells with clean backgrounds. But we also realize that the number of CTC, the presence and absence of CTC did correlate with the disease progression. But I also realized that the number of CTC is too low. And because we do not do the enrichment, because the assay is highly specific and sensitive, which makes me believe that the clinical utility of CTC is likely to be limited. And I decided to completely stop the program. And that's when the CTC field just heating up. The CTC field was really hot in 2010 and 2015. Hundreds of millions of dollars, hundreds of companies have started. But that's when we already retreated from the field because we realized that the clinical utility are likely to be limited. The biology is not cooperating. And looking back, that's probably one of the most important strategic decisions that we make. If we put 100% of our effort into CDC, the company probably would have long debt. But still, the company is considered a success story by SBRR and become one of the highlighted company. And the reason is that we did use the funding to develop groundbreaking technologies which form a solid foundation for the success of the companies. And so the pivot that we did is to tissue RNA in situ hybridization. As I said, the RNA in situ hybridization technology started in 2069. But over the last several decades, the market never took off because the technology do not have sensitivity, do not have specificity, and workflow extremely complicated. But there's still a lot to be learned about the spatial biology, about cell and tissue and tumor heterogeneity. And also immunohistochemistry assay is difficult to develop for many of the targets. So RNA potentially could be a great surrogate to develop RNA in situ assay in place of immunohistochemistries. So when I look at the market opportunity, I also believe this is likely to be a very large market opportunity simply because the neighboring market, the immunohistochemistry market is $2 billion and the DNA in situ is $500 million market. So with that conviction, we decide to put all our effort into developing a tissue in situ hybridization technology, make it fully automated, make it easy to use, and make it extremely robust and high performance. And as a result, now it become a gold standard technologies. And this is some of the images that we got. And I want to mention that when we sort of got the technology work, one of the first collaboration I had is with Lin Mei's lab, where we published, I think, the third paper using the RNAscope technologies. Now it's over 6,000, as I mentioned. And recently, Biotechnique celebrated 40,000 probes using the technology by a variety of different labs for different tissues, different genes, and different species worldwide, celebrating the fact that we have pioneered spatial biology and the RNAscope has become the gold standards. And looking back, maybe there's a few key factors for startup success. And first, I think is that you need to identify a significant problem to solve. And second, you need to come up with a differentiated solution. And third, you need to develop conviction in your idea and willing to bet your whole career in it. And last, when the situation demands, you need to make the right strategic decisions to make changes as necessary. And of course, there are other factors that also contribute to the success of a company. Luck is also as important as other factors. If I don't get the support of the grant reviewer, you need 3 grant reviewers, all like it, to get funding. I would have to ask my wife to approve to fund the company. And if I don't meet the Dr. Gerald Chen, I don't know whether I will get Series A funding or not. So luck clearly also play a role. Maybe last slide, I'm highlighting acknowledging mentors. And I couldn't be where I am today without the support and advice of mentors, advisors throughout my careers. I'd like just highlighting a few. And first is my PhD advisor, Ken Neat, who taught me about how to do solid science and how to be humble. Unfortunately, Ken Neat passed away a few years ago due to melanomas. Second, I'd like to acknowledge Mark Tassi-Levine, who introduced me into the biotech space and referred me to my first startup, Exelixis. And at the time, Mark is a professor at the UCSF who worked in the same field as me and appreciated my work. Now he's the president of Stanford University. I also like to thank Frank Whitney, who was the CEO of the first company I co-founded, Panomics. I learned a lot from him about business as well as how to be a leader, and also greatly appreciated his support when I started the ACD. And he taught me that always over-communicate, not under-communicate. And last but not least, Gerald Chen, who took a chance on me and took a chance on ArcScope technology when it's at a very early stage of the development. Gerald commented that ACD is a textbook example of venture-funded companies, and for which I'm extremely grateful. Thank you again, Dean Gerson, for your invitation. It's such a privilege and honor to give this lecture. Thank you.
You and I'll leave it up here, but there'll be plenty of good ideas that you can put into this bowl. So this is distinguished Dean's Distinguished Lecture, Yiling Liu, PhD, 1992 biochemistry. I'll leave it up here.
Thank you.
I'm going to ask Jay to come up and you guys move over here and sit down. We'll take some questions and comments from the audience. And I think we all appreciate that if this is to our graduates, we're doing quite well. But please ask away. I have some questions if I need to, but I'd just soon heard from all of you.
I'd be curious from each of you. I'd be curious from each of you of what you think the role of a synergistic community was as you began your first companies. And I say that because most of the places in the United States wish they had a large synergistic community, hope to have a large synergistic community, and clearly the two of you have taken advantage in your careers of places that already had large synergistic communities. Would you comment on how important that was?
I can start. I mean, ironically, living in San Diego, we thought that we were underprivileged relative to San Francisco and Boston and New York. And it is a continuum. We didn't have the venture capital depth. We had to go out and get our money raised elsewhere. But so in saying that, I'm acknowledging how important it really is. The fact that it was less in some ways made the fight harder. You had to do more. Of course, the advantage of San Diego is you have the Salk, UCSD, Burnham, other schools, all within a relatively small distance of each other. And there's a huge advantage when you're recruiting and hiring people. Very quick, there was high turnover, of course, because of that, but huge advantages. But I think the capital aspect of it is also important. And San Diego, even today, is just now getting the point where it had enough of those larger companies and those larger success stories to be able to access more capital there. But it is a factor, and I think it just increases as you get away from certain pieces of centers, and obviously we took every advantage of them we could.
Yeah, I totally agree with you. Clearly, the community is important, both near the vicinity of the company as well as far as well. Right? In our case, we're lucky in the Bay Area, so we have access to talents from UCSF, UC Berkeley, and Stanford universities. We also have a lot of biotech companies, And I was lucky that my ACD was incubated with my previous company, where my CEO, Frank Whitney, gave us a lab to test ideas, right? And also from afar, clearly the grant support was very helpful, greatly reduced the risk for me and my family. Yeah, so community is clearly important.
So will Zoom
And we had a
reduce that?
collaboration. Sorry,
Yeah,
we had a
no.
collaboration with Cleveland Clinic in my last company. Published some of the first papers in Science and others. So community is important. Yeah.
I just was saying, I wonder if Zoom and other ways that we're collaborating could help reduce that limitation and island feel, and maybe propagating that, because now we have employees all around the United States, and it's a different world.
There's some folks that you know well. Go ahead, please. Yeah, there you go.
Thank you for so inspiring career achievements. One of my question is, so when you're a student or you're a postdoc, you know some of the concepts, science, and techniques, and you believe you have a great idea. And the question is, how do you make that to move to actually get a marriage with a business person, with your CEO, and make it successful? Because it's just limited to your own knowledge, skills, and idea may not take you too far. I think that the next step to make that bridge between the business world and academia, that's what make it the key ingredient to be successful. So how do you make that bridge?
Yeah, for me, I think it starts with the unmet need. I think first of all is that will you be able to identify a significant unmet need, and secondly, can you come up with a solution that's differentiated? Do you have that insight, the kind of solution that you truly believe? So when I usually come up with ideas to solve one particular problem, whether it's my last company, ACD, or this company, Alamo Bioscience, I myself will spend a lot of time to think about how to defeat the idea. To say, what could make this idea not working? Until I think all the possibilities of trying to defeat it and couldn't shoot it down, then I will start to develop the conviction to say, okay, this is the idea that worth pursuing. And then are you willing to take the risk to put all your effort into it?
So necessity is the mother of invention can't be more accurate. I mean, I think understanding that need and solving that problem matters. Translating it, though, when you're a professor or a postdoc and you aren't in the industry, depends. You may not want to take that leap. I think then a lot of people I'm familiar with have aligned themselves with somebody that's already done something or wants to do something, because sometimes the passion of wanting to do something is more important than the having done something. And I think there are a lot of people that have, if you think about most companies, most are forged through collaboration or technology coming out of academia. It's kind of rare for the companies to create their own technology and develop it. But so to me, echo 100% what you said, but I think then how do you execute to the next point? Sometimes that requires some help if you're not willing to jump in it yourself or the circumstances don't allow. But that doesn't mean you should be shy of doing it at all. I think most companies have come up that way.
I think it's very important as an entrepreneur that you need to be open-minded and learn from people who are more experienced, and getting all the perspectives that you can get, and make a decision based on your own situation, because nobody knows about your own situation than anyone else. But it's important to get those perspectives from the different people that you get, whether it's on the business side, on science side, or technology side.
Lynn? So I wonder what you can tell us. So we do basic research, and occasionally we come up with the translational ideas, and we file disclosure and get patent, and we just wish the patent will be licensed, but many of them won't. But some of us at least, or I believe some of our patents, have true value. What can we do? Should I quit and become 100% devoted, seek for money, set up a company? Looks like you two being successful, but you are so dedicated and to what you believe. But at the meantime, we have to teach, we have to write grants, have a lab to run.
Yeah. I think you got to have capital, and that's something you have to think about, because it isn't just wanting to do it. It isn't that it's just a good idea. You got to say, if I'm going to jump, how am I going to get from A to B? How am I going to survive? With Bioatla, we decided, as I said, that we understood enough about antibodies. We were confident we could make technologies for it. And we had just enough capital to fund for a while. And we said, we'll do services for other people. So the first year of revenue was $5,000. So it was obviously a year of just spending money. We did hire good people. In the second year was over $1 million. And then it went up to $5 million before, by the time we had figured out the CAB and PAX technologies and said that we were going to wind out of services. But it was scary because the revenue went like that, and you really have to maintain costs. So to me, the capital piece of that and understanding how you would form that will inform what you want to do depending on your own situation. But if you haven't got a path to that, that's hard. You're really jumping into the abyss in a way, and it's very difficult to manage if money runs out. And the pandemic almost wiped out Bioatla. We were really close, and wholly unexpectedly, because no one saw this coming, and they quarantined everything. So you really do need to understand the capital paths.
Yeah, that's the difference between high-tech and biotech, right? So high-tech, maybe a few computer does enough. But high-tech, the biotech is a laboratory-based medicine. You do need to have significant findings. One way is to convince VC when the idea is still early. But the other way, which I have gone through, is the government grant. Even though for my Alamo Bioscience, I don't need to write a grant because I have my own funding to do it, I still have the habit to write a grant, and got the grant out of it. It's just kind of fun to test ideas, right? So that certainly is one of the things that you could consider, write an SBR grant to get started.
I'm going to keep Jay and Yu-Ling around for the next half hour, 45 minutes. We have a nice reception outside, and I both would love to continue the discussion in smaller groups, and they're here as your advisors for the afternoon, guys, so I'd ask them the questions. Alan, do you want to ask one last?
I have one more, thank you. One more question to follow on the last point that both of you were raising, and that is there are many universities, let's just talk about the United States for a minute. There are many universities, some of which are very pleased with their faculty, their students, their trainees go off and start venture opportunities. There are some that embrace it even more, where they bring companies into the university campus. They allow them access to all of the resources of the university. You know, they have to set up the various rules and costs and so forth. Would you comment on that model, the latter model?
Yeah, I would definitely encourage universities and the faculty to think about translation of their research, because I think if you look at the institution like UCSF and others, they have hundreds of millions of dollars coming out of the royalties of the patents, right? And a lot of this is conceptual, where the faculties, they do not know that, you know, some of their work potentially has translational value. So part of that is probably, you know, some education about faculties, you know, because I think West Coast and East Coast, Harvard or UCSF or Stanford, they just have much more exposure about the companies, the entrepreneurship. Maybe in the middle of the country, there's less of exposure. I think they could benefit from those trainings and exposures.
Yeah, I would echo that too. I know Scripps and Burnham both are pushing very strongly on this translational side. They still have the basic research side. I mean, they still can win Nobel Prizes, but they really are pushing translation. I think it's pretty important. And I know that even with UCSD, when we were doing ADCs, we were doing confocal microscopy, we were able to lever the center at UCSD and pay them to allow us to do our early studies. It really helped the company not have to invest in that equipment. So it really matters, and you create relationships. And UC system is one of the hardest to deal with, but I think the universities around that have done quite well on it. I think it's important. And I think about the engineering field, I think it's always been applied, but you know, biology is getting more applied, and it's got huge mountains to climb, and I think you need it personally.
Thank you.
Great. Thank you very much, Jay and Yuling.
Thank you.